Okay, so today I will talk about a paper published in Neuron in 2017. The paper is called High Resolution CBV Functional MRI, a low mapping of laminar activity and connectivity of cortical input and output in human M1. So human M1 here, you want to measure the gray matter activity. So we know the gray matter contains 6 laminars from layer 1 to layer 6, from the superficial close to CSF to the deeper tissue close to white matter. So as we know, cortical actually can get some cortical to cortical input from layer 1, not layer 2, layer 3, and also some thermic input from these 2 layers. And after some computation, it can get some output to control like the muscle to pinch or to kick a stone. The signal come out from the layer 5, layer 6 of the M1 and through the CST, that is cortical spinal tract, to the alpha motor neuron in spinal cord. Okay, so if we have high resolution signal, and then we can use GLM to get the input and output in layer 2, 3 and layer 5, 6. So the author used a sequence called the SSSIVSO sequence. This is the full name is called slab saturation, slice inversion, vascular space occupancy. So in this sequence, he can saturate the blood signal from the whole slab, and using the inversion technique, and to only make the magnetization in the imaging slice steady state, and the signal in that slice is high. So when you assume that both the blood and the tissue actually they have the same decay, that is tissue star decay, and then you can calculate the CBV. So I won't talk about this in detail. You can refer to another paper published by Huber. But using this method, he can measure both task functional MRI and the rest state functional MRI. So for the task MRI, he invented 4 tasks. The first one is typing with touch. You can see this figure. Figure A, you see you use the right hand, and with your forefinger and your thumb you can tap, and during this procedure you can also get some feeling in the S1 region because you touch, and because you tap, and you need to output some signal from your M1 to control the muscle. So expected you will get 2 signals, both from layer 2, 3, that is S1, and maybe premotor can give some input through these 2 layers, and also you can get signal from layer 5, 6 because it sends out some control signals to the muscle. The second task is typing without touch. So in this case you try to tap, but without touch. That means the sensory S1 there is no signal, so there is no signal input from S1, and the layer 2, 3 the input signal actually is going down. The third task is touch only. That is a passively touch. That means you don't want to control your muscle, but some other things get contact with your forefinger and the thumb. So you still have S1 signal input, but you don't have output. The last one is typing with left hand. That is contralateral hand. That means your contralateral M1 will be activated, and that area can send some signal to your left M1. So after using these 4 tasks, we do the measurement for each task, highlighted in different colors. So with CBV, we can see the blue curve, that is for the tapping with touch. You see there are 2 peaks in both layer 2, 3 and layer 5, 4, 5, 6. And if you use the second task, that is only typing without touch, and then you will see the S1 signal from layer 2, 3, the signal get down. And if you use the third task, that is only touch, and you will see there only S1 signal, but without output signal from layer 5, 6. And if the task 6, the epilateral typing, and you will see the counter hemisphere M1 actually send a repressive signal to your left M1. This is negative, the red curve. So the author said all these tasks actually can reproducible get this signal pattern from across all 11 subjects, and this is the average final result shown in this the figure B. You can also see the bold signal actually do not have the double peak pattern. They all monotony decrease as the depth goes down to wet matter. So for each subject, actually, you can measure the signal from layer 2, layer 3. Then he put it as x-axis, the signal strength, and he put the signal from layer 5, layer 6 as y-axis. And then for each task, you can have actually have 2 measurements and with 1 scatter point. So for each subject, you have 4 points on the plot, and each colored in different color that represent different tasks. And so from the CBV cross plot, you can clearly classify different tasks with the measured 2 peak signals. But using bold, you cannot differentiate between the tasks because they do not have the resolution. Especially the bold actually can be the signal can be influenced by the joining wane. The joining wane actually make the 2 peaks actually disappear. So he also used the method in red state functional MRI analysis after acquiring the data. For each also actually you can have a time series, and then you can first you see here this image. It shows that this is precentral cortex, and this part actually is M1. M1, it contains the deeper layer that is supposed to be the output activity region, and the superficial layer in green actually is the superficial layer that is supposed to be the input from some other cortical region. So if we put a seed in the deeper region, and you will see the M1, other voxels in M1 actually can co-fluctuate with the seed. At the same time, the premotor voxels can also fluctuate with this region, with this seed. So that means the premotor actually have some correlationship with the deeper layer of M1. But if you put seed into the superficial layer, for example, you randomly select one voxel in superficial layer of M1, and you will see the M1 region other voxels can co-fluctuate at the same time. The S1 voxels also co-fluctuated. And then if you put your seed in S1, and you will see the M1 superficial layers have some voxels co-fluctuated with your seed. But if you put your seed in premotor, you will see both superficial and the deeper M1 voxels can co-fluctuate together. So that means from S1, you can send some input signal to M1, layer 2, layer 3, and but you cannot influence the M1 output from layer 5, layer 6. But for premotor, it can both modulate the M1's output signal from layer 6, layer 5, but it can also give some input signal to M1's layer 2, layer 3. So this is the final result, and in sum, the author want to say the CBV-based functional MRI that can sample activity across layer or across the depth or across lamina. And the CBV-based functional MRI have higher resolution than both functional MRI, so it can separate the measure input and output activity. And for the resting state functional MRI, it can reveal the directional connectivities.